Takes just a second here. Okay, physiology of the kidneys. Here you have another gross picture of the structure of the kidney, the nephrons, ascending, descending, and ascending loop of Henle, Bowman's capsule, proximal, distal convoluted tubules, and the collecting ducts. So you need to know what happens in each of these areas. You need to know kind of why, or you need to know why the countercurrent multiplier, what it does and how it works. Kidneys, important functions. Their osmotic balance. You have the baroreceptors. Maintenance of pH, excretion of hydrogen or reabsorption of the hydrogen, as we've talked about. Body fluid levels. The kidneys, by how much water is put out in urine, can control the body fluid levels and the excretion of toxic waste. You need to urinate 500 to 600 cc's a day just to get rid of the toxic waste. You can find various things in the urine that may indicate health or a lack of health. Liver problems, you're going to see bilirubin. Diabetes, you're going to see sugar. Things like that. The urine volume and concentration are regulated by hormones, and two of them. One is ADH, the antidiuretic hormone, and the other is the renin-angiotensin-aldosterone system. ADH, as you saw earlier, works on the distal, on the collecting duct, allowing aquaporins in if you are dehydrated and you don't want to be excreting urine. The ADH allows aquaporins. It allows water out of the collecting duct into the interstitial interstitial area, picked up by the vasa recta, dumped into the inferior vena cava, and back into circulation. The renin-angiotensin system, aldosterone system, regulates the reabsorption of potassium and sodium. Actually, it regulates the reabsorption of sodium, and it draws potassium and chloride back in with it. In some of the diseases, you see an aldosterone secreting tumor. The sodium is very low. The potassium is very low. The sodium is very high. Okay, by drinking large amounts of water or consuming salt, which is what we did in lab, you can cause the body to change the volume and composition of the urine. You saw that for those that drank the tomato juice versus those that drank just water versus those that drank the caffeinated sodas or just the non-caffeinated sodas. A measure of the solute in the urine is the specific gravity. That's the amount of dissolved solids. Normal is 1.003 up to 1.030. There is a disease with the lack of the cortical hormones called diabetes insipidus, where you urinate a tremendous amount of water and the specific gravity is right around 1.000 or 1.001. Excuse me. Now you're going to look at various abnormalities in the urine. We did various tests on the urine. Obviously the pH. You're looking at the pH, the amount of hydrogen ion excreted by the urine. The urine can only concentrate down to about a pH of 4.5. After that, it's beyond the ability of the kidney to excrete that amount of hydrogen. You're going to look at the glucose to see if glucose is spilling over. You have glucose when your glucose gets above about 140. When the glucose gets above about 140, it spills over in the proximal convoluted tubule. It cannot absorb that much more glucose, so it starts to spill into the urine. You see this in diabetes or people that have been given a large load of glucose. You're going to look for ketones. the byproduct of protein and fat metabolism. That, along with the positive glucose, may indicate a ketoacidosis, which is a very dangerous condition for the patient. If you're going to look at protein, you should have no protein in the urine, or very, very little, I should say. And if you see a lot of protein in the urine, you're looking at some sort of renal failure or damage to the nephrons, via whatever. It might be a bacteria, it might be a chemical or trauma. You're also going to look at, let's see, we did pH. Hang on 1 second. I'm sorry. We did pH, we did glucose, bilirubin if you have a kidney problem, a liver problem and like a hepatitis, it's not going to process, you're going to be excreting bilirubin in the urine. The specific gravity, again, if you're dehydrated, it's going to go way up. It's an indication of the hydration of the body. Protein, you shouldn't be. You look for nitrite and leukocyte esterase in a kidney infection or your bladder infection. Those are from the white cells and the nitrites produced by the bacteria. So that's about it. Blood, you should see no blood in the urine. Sometimes after being very warm, trauma, heavy exercise, things like that, you will see a little bit of blood in the urine, but it should go away very quickly. Kidney function, regulation of extracellular fluid, blood volume, waste, electrolytes, pH. Urine is made in the renal pelvis, down the ureter to the bladder. Through the bladder, through the urethra, exits the body. There's a kidney structure. Two distinct regions, the renal cortex and the medulla. Medulla is made up of the pyramids and columns. Each pyramid drains into a minor calyx, major calyx, renal pelvis. Micturition is the ability to urinate when you want to, and we're not going to go into that. Kidney structure, the nephron, again, functional unit of the kidney. Renal blood vessels, we're not going to go in other than to know that the vasa recta and the capillaries are excreting in the Bowman's capsule and reabsorbing, and you have the afferent and efferent arterioles. Nephron, made up of Bowman's capsule. Filtrate goes in from Bowman's capsule into the proximal convoluted tubule, into the descending and ascending loop of Henle. After the loop of Henle, fluid passes into the distal convoluted tubule, finally into the collecting duct, and then starts its drainage process. Two types of nephrons. You have the juxtamedullary and the cortical. I'm not going to go into that. Glomerular filtration. Capillaries are fenestrated. They're large pores. Large pores allow solutes to leave, but not the cells or the proteins. Okay. This you can look at in your book, glomerular capsule. We talked about that. Ultrafiltrate. Fluid in the glomerular capsule gets there via the hydrostatic pressure, and then collects in the Bowman's capsule and moves through the proximal convoluted tubule, the loop of Henle, distal convoluted tubule, collecting duct, etc., etc. Glomerular filtration rate is the amount of filtrate that can be produced. Somewhere around 180. That should be liters a day, not milliliters per day. Renal autoregulation, the GFR is maintained by the blood pressure. I'm not going to go into that as part of the kidney. Just know what the counter current is. Know that as the filtrate moves down down the descending loop of Henle, water is drawn out by osmosis. The pressure gradient is maintained by the sodium pumps that is actively transporting sodium out of the ascending loop of Henle, what water cannot get out of, but it creates a heavy gradient osmotic gradient that pulls water out of the descending loop of Henle and is reabsorbed via the vasa recta and back into the blood system. Going on with the lab, we're going to look at the urine under the microscope. You're going to look for casts, red blood cells, white blood cells, and bacteria. You usually always have some bacteria in there. When you culture, you have to have 100,000 per milliliter before anybody worries about it. Some of the colors in the urine: red to dark brown, hemoglobin; brown to black, melatonin from a melanoma; wine, dark wine is a jaundice; dark brown is a hepatitis. Glucose you'll get in from stress. Remember, you're looking at the sympathetic. Go back to the sympathetic nervous, and the fight or flight syndrome causes your glucose to go up. Sometimes if it gets above the 140, it spills over into the urine. If you see leukocytes, you may have a bladder infection. Same with erythrocytes. CAS is a glomerular nephritis, an inflammation of the glomerulus, allowing protein across, which forms casts in the tubules. Crystals, you may have kidney stones, gout, hyperparathyroidism with calcium. That's pretty much what we looked at here. So you took the kidney, took the urine, you did the filtrate. Not the filtrate, you do the dipstick, and then you used Sternheimer-Melvin stain to do to look at the cells. And that's about it for reabsorption.